Thank you all for coming here today and for providing me for the opportunity to speak about our lab. Um, our lab sort of focuses on trying to understand disease using high throughput technology, computational approaches as systems biology. And basically we do this sort of the whole thing. We traditionally lack focus. So we start with profiling of human samples, applying computational biology and animal and cell culture models and verifications. So it's the whole sort of the whole gamut of translation. When we the disease we mainly study is idiopathic pulmonary fibrosis. It's a chronic progressive form of lung scarring. It's defined by exclusion of all known lung disease. Around 200,000 people in the US, 6 million worldwide. The incidence increases with age. So at certain ages, over 65, it's more common than most cancers. Extremely rare below 55. Median survival is actually 3 years. So it is a really deadly disease. If we were able to prolong survival by either 20 or 30%, we would have, this would become a really common disease. We have now 2 FDA-approved drugs that slow down progression, and there is a very strong genetic background. So around 30 to 40% of cases are associated with genetic aberrations. Just to show you that we are very active. This is, I like numbers, and what I calculate is basically all the papers in PubMed from 2002 till now. And basically the black line is cancer, the dashed line is PubMed. It's all normalized to background. And you see that cancer sort of drives the numbers of publication in PubMed. But look at IPF, this is the red line. We basically increased the number of publications by around 600% over the last 5 or 6 years. And much of it was driven by the discovery of the genetic associations and the application of high throughput technologies. So this is a really hot field, a lot of discoveries, a lot of high impact papers. What happens in IPF, this is the typical lesion in the IPF lung. To those who are not used to look at histology, on this side, you see normal lung, and adjacent, you see a myofibroblast focus. And what we're interested in is to actually understand how the lung sort of gets messed up. And this is sort of the pulmonary fibrosis GIF. The reason I'm showing it to you is because you need to know that I can make GIFs. And basically, the whole idea is that you have the normal alveolus, normal lung, then there is recurrent injury, a change in cellular interaction that leads to pulmonary fibrosis. Of course, these are really nice words, and we can say we have a model, but actually, this model really does not tell us what's going on. So what cells should we target, what pathways? And my group has applied mostly high throughput technologies. And just as an example, this is sort of the projects that are available for computational students to get involved in the lung. There's a whole group of people doing single cell and systems biology work. The IPF cell atlas, where we're resolving lung, blood, immunophenotyping of the lung, the normal lung aging cell atlas, the pulmonary fibrosis connectome, I'll mention it in a second. And we have been involved together with several other groups, mostly with Charles de la Cruz and David Huffler's group, and Kiko with the COVID-19 cell atlas. We have a really cool multi-omic paper that is really worth looking at. And then there's new therapeutics. My group has discovered several therapeutics. I'll just mention again, we have a new thyroid hormone agonist that seems to protect the lung in both fibrosis and COVID-19 acute lung injury. The last thing that, again, relevant, especially to people interested in networks biology, is the role of microRNAs and large encoding RNAs. I won't go into detail, but there's plenty of projects. So this is the IPF cell atlas. I'm showing you this beautiful three-dimensional UMAP that shows you basically this is every single cell type in the lung. around 328,000 cells from 70 individuals. And the idea is that we try to understand molecular mechanisms of disease using, for instance, dissociated human lungs. And we also generated a freely available website, and we're leaders in creating these kind of fields. If you raise your iPhone, scan your screen, you can actually have access to the IPF cell atlas very easy and very accessible. So what we discovered most recently, again, this is new maps of all epithelial cells in the lung, and we identified this unique population of cells only showing up in patients with pulmonary fibrosis that have very, very unique characteristics. Basically, they are cells that sit at the edge of the injury and probably drive the disease. And then we call them aberrant basaloid cells. So this is one example. Now, this is a lot of biology, but this is a lot of computation. There's no way you can identify these cells if you don't apply advanced computational approaches and statistics. This was published recently in Science Advances. We also identified a novel endothelial cell population and verified its existence. And we identified, like many other people, what we call profibrotic macrophages, so a unique population of fibroblasts, of macrophages that have specific cell surface markers. And we identified them all in multiple data sets. So again, I'm showing you results, but all of this work requires computation, visualization, statistics, systems biology, data mining, everything that would be ideal for training. Because the lung is really characterized by spatial and temporal heterogeneity, we do also work on image analysis, and we try to understand what is going and how the disease progresses within the lung. And most recently, we developed a dynamic regulatory network explorer that actually, this was in collaboration with collaborators at CMU, that allowed us to look at patterns of gene expression within the individual lung. So we take one lung and look how disease progresses. And one of the things, and this was recently published in JCI Insight, and again, the data is on the IPF Cell Atlas or on the CMU's website, is basically we can identify distinct regulators that occur at early or late events in pulmonary fibrosis, and we hope to develop this for fibrosis. Of course, because fibrosis is actually not a disease of a single cell, it's a disease of an organ, we want to understand how the different, how gene expression regulation and cell interactions regulate the phenotype of an organ. This is a project sort of in the beginning of the thinking about it, but in this we applied an approach to gene regulatory networks that allowed us to look at sort of the cell gene expression regulation in control lungs and IPF lung, identify distinct areas. And then what we did is we actually looked using the Broad Institute's perturbation map to compounds that can target different distinct areas in the network to identify potential drugs. This was really cool, and we found some really cool stuff that's actually driving some of our work. But what's more important, it led to a really ambitious initiative that's going to start in my lab this year, which is called the Pulmonary Fibrosis Connectome Project. And in this idea, what we do is we basically will do a perturbation map of single cells, organoids, and other translational models in humans, and then try to see what fits the human lung. Trying again to identify, are there agents that have been approved that are not dangerous, that we could use to try to normalize the lung as an organ and not necessarily target one pathway or one cell. And the aim of this project is really to reverse fibrosis. So that's who we are. Again, we're a sort of a combined mixed lab of heavy duty genomics, high throughput biology, medicine, molecular biology, and computational biology. Usually when students come to my lab as computational stats students, they have other mentors, and I share students with Dr. Hong Yu Zhao and with Joseph and Carnegie Mellon and other people, um, and um, and also Yuval Kluger and others. So the whole idea is that we create sort of a mixed methods, inter and multidisciplinary mentoring team, which I think is a lot of fun. So this is a picture from last year when at the end of the year, people come and leave and we take a picture and we give them a picture of previous years. Unfortunately, this year, as you all know, it's all by Zoom, but hopefully next year we'll be able to, maybe the end of next year, we all are able to get together. So we'll stop